we're not trying to attempt to replace all these existing repositories. We just want to kind of expose their content and make it easily searchable, as well as kind of following FAIR principles as best we can to make things findable, accessible, interoperable, and reusable. So we started WorkflowHub a few years ago now, during the COVID-19 pandemic. So it was originally part of EOSC Life. Now it's kind of funded by various things. We'll see some logos in a bit. But it's very much a community-driven project. So we have regular community calls. It's kind of managed by a very international group of people. So we've kind of got the UK and Belgian nodes of Elixir, but also down under we have people from the Australian Biocommons who are very much core participants in WorkflowHub and the team. So yeah, as a brief overview of what's happening in WorkflowHub. So we've got over 500 workflows now. Over 26 workflow systems are supported. So actually WorkflowHub is agnostic to workflow systems. We have dedicated support for some of the more popular ones, such as Galaxy, Nextflow, SnakeMake, CWL. But actually you can submit a workflow in any format. And we have, over 600 registered contributors. That's people who can kind of contribute content to WorkflowHub. And they're distributed across over 200 teams. And we're also getting a lot of collections now, which are kind of curated sets of workflows around a specific activity or theme. And yeah, what the workshop today is about the SOP support. So SOPs, SOPs, protocols, all the same thing. A little note about WorkflowHub is based on the FairdumSeek platform, which is a data management platform. It was originally produced to kind of serve the systems biology community, but now it's kind of cross-domain. And fairdumhub.org is the main instance of that, which you may have heard of. And WorkflowHub is kind of repurposing that software to act as a workflow registry. But we also switch on various features of that software, one of which is being the SOP support. So you can create SOPs in WorkflowHub like you can workflows and collections, etc. We don't have a ton of SOPs right now. There's only 5. It's just a quick screenshot showing you the current state of the art, but hopefully when we get some good ideas after today's workshop, we can boost those numbers a bit. So what can you do with SOPs and WorkflowHub? Well, it's very similar to what you can do with workflows, basically. So we support SOPs in various formats. Any format, basically, you can upload. You can also register remote SOPs. So maybe you've got a Google Doc or something that describes your protocol. You can link that. Obviously, we have an array of metadata you can provide to give it appropriate credit, tags, and specify what the license is. You can currently link workflows to SOPs. Maybe, a workflow is following a certain protocol or something, so you can link those workflows to the SOP. They're versionable as well, so maybe you came up with a new version of the protocol, you can upload that. You can mint a DOI for an SOP to make it more easily citable in papers, etc. And yeah, we've got the usual search and filtering based on the metadata that you've provided. And you can kind of organize things into teams and collections like you can do for workflows. This is just I think this is actually an old slide from the BGE meeting, but this is what we're going to do next with SOPs in WorkflowHub. So obviously here we've got a point about collecting requirements, and that's what we're here to do today. Some of the things we've already done, such as linking SOPs with workflows and the tagging and filtering. So yeah, I'll just plug the Workflow Hub Club, which is the kind of community meeting around Workflow Hub. Happens every 2 weeks. So the next one will be on Wednesday next week. And that's kind of where we discuss all the happenings in Workflow Hub and the surrounding workflow community as well. So there's lots of people who join that, talk about fair workflows in general, papers that are being written. There's lots of discussions about RO-Crate if you're into that. So there's a link there with how you can get involved. And we also have a Slack channel where we also discuss Workflow Hub and other workflow activities. Yeah, these are just some of the acknowledgements for the people and groups who have participated in making Workflow Hub what it is today. So a lot of those are actively funding Workflow Hub now or have funded it in the past. Yeah, I think this is going back to Nick's slides now, so
yeah,
I'll stop here unless there's any questions.
Yeah, I think we've got time for one question if anyone has one.
Sure. Let me know because I can't see the chat.
Yeah, I don't see one appearing. But on the subject of RO crates, I think probably we'll have one similar workshop to this on RO crates in the near future. Okay, thanks, Vin.
Right, cheers.
If Ben, are you ready to go?
Yeah, let me just share my screen quick.
Great.
I need that to be enabled again.
Okay.
Right now.
Did that work, Ben?
Yeah, we're getting there. My computer's slightly slower than me.
Okay.
Right. Can you see that, Nick?
It's loading. Yes.
Okay.
So,
and hopefully
thanks.
it's now gone full screen.
It's gone.
Okay.
Great, thanks.
Hopefully my internet situation improves over this talk. But yeah, I'm going to give a little overview of some of the SOPs and protocols that we use in the bioscan stream of Biodiversity Genomics Europe. And I've seen a couple of my colleagues from that project on the call, which is great because they can feed in when I get things wrong. So yeah, my name's Ben. I work at the Natural History Museum in London, and I'm talking about BGE. It's a giant EU-funded project, which takes two continental-wide networks and joins them together. So ERGA focused on genomics or genome sequencing, whole genome sequencing, and Bioscan Europe focused on DNA barcoding and metabarcoding. And there are over 200 organizations in that network, but around about 30, I think, in the project BGE in particular. It's a giant project, and the overarching mission is to accelerate the use of genomic science across Europe. And it's sort of divided into three sort of sections, building capacity, and this is where SOPs, I think, in particular come in, where we can have training and documents that support that training, and then using that capacity to really get production scale genomic exercises and investigations happening across Europe, and then turning that data into something useful. And the way the project is organized is in two streams. So on the sort of middle there, you can see the greener eyeball Bioscan Europe stream, which is what I'm mainly talking about today. And then in sort of pink red, you can see the ERGA EBP stream on genomics. But they're arranged in a sort of stream and pillar approach, very clever, where we have work packages for node development, sampling, sequencing, data, and applications. I'm the sequencing work package lead, but I'm heavily involved in work package 4 at least, and work package 8 as well. So the sampling, sequencing, and the data side of things. And then, so if we look at the Bioscan stream in a little bit more detail, it just starts with training on the left-hand side there. and then again, sample sequencing, data, and applications. And we've got it split between single specimen barcoding and community or environmental DNA metabarcoding. And you can see there's a lot of activities happening in each of these little blocks, and many of them require protocols or SOPs. And we currently use a mixture of Google Docs. If you're lucky, that gets turned into a PDF. Otherwise, for some of the lab stuff, we use Benchling. For some of the protocols in the lab, we've put them onto protocols.io, but we've only kind of just started doing that. And then for the data related stuff, it's an active development at the moment, so it's mostly in GitHub. And so I'll go through from that left training through to the data and just give you a few examples of what we're doing in BGE. So one of the big things is around training. And so we did a workshop on how to do DNA barcoding and the analysis associated with that around using Oxford Nanopore Technologies. It was hosted in the wonderful Thessaloniki in Greece. And as a result of that workshop, we're building this handbook. And you can see that this handbook has 16 chapters, and it's essentially how to prepare and deliver a workshop. But each one of those chapters is quite specific and could probably be its own thing that has multiple versions over time, especially the lab things and the data things. Those are going to change sort of monthly almost these days. As part of that training, when we were doing lab stuff, we were using Benchling, which I mentioned earlier. But I think Caroline will be talking about much better implementations of Benchling. This is just where we have our sort of lab protocols, and you can link them together if you're working on samples and things like that. When it comes to specimen data, it's essentially Google Drive or Google Docs that have been turned into PDFs. In this particular case, we've got a metadata sheet for specimens that you can see on the extreme right there, just a bit of it. And associated with that spreadsheet is a document that says, okay, for this field, we mean X, Y, and Z. This is what you need to put in there. It's the colors and the formatting are wonderful. And then that is just really a subset of the wonderful Barcode of Life data systems handbook. So this is a sort of annually updated, I would guess, handbook as they build their database, the BOLD database, and it goes in extreme detail onto everything you could possibly do with their database. But it's updated as a sort of PDF, I guess, once a year or whenever there's a significant update. What we did for BGE is we took the bits that were super relevant, and we, of course, wanted to customize it and add extra fields. So we added that to our SOPs. Tissue sampling, again, this is it's PDF based, and we were very lucky that the eyeball Barcode of Life people have a tissue sampling SOP, so we were able to repurpose that and edit it to fit our needs. In particular, because we're doing a lot of genome skimming, we had to change some of the text here around lab cleanliness and tissue amounts and things like that. But it's a sort of pictorial with step-by-step instructions for everyone on how to fill out plates and the data associated with those plates. Field sampling, this is just, again, it's how to do eDNA filtering in the field, and it comes as a PDF or a Google Doc, and you with step-by-step instructions. But again, these sorts of things do change over time, and we don't really keep track of versions at the moment. When it comes to the DNA labs, which is kind of more closer to my wheelhouse, we are using protocols at the moment, but we haven't put a lot of effort into getting all our protocols onto protocols because it does have like sort of import features from Word documents and things like that. But there are aspects of it I don't really like, where the steps are so far apart visually, and what you see in like a QIAGEN kit where it says step 1 to 10 suddenly takes up a whole screen or something like that. But anyway, we've been putting some of our protocols into there. And the good thing is they get DOIs, they're versionable, citable, and all those sorts of things. It's really handy for, I guess, molecular techniques where you need to be able to see quite quickly volumes of reagents and things like that, and you can change them and adjust them as you go. And then moving on to the data side of things, yeah, GitHub. I'm not going to labor the point here. We just use GitHub at the moment, but we try and make sure that the things we're developing on GitHub have got lots of information about how to set them up, how example analyses and things like that, so that people can run through them. This is the sequence assembly pipeline that we're working on at the moment. And then there's another one which is around the the very right-hand side of my starting screens are the data in the application. So this is how you would create and curate a reference library for European species. And so that's got an SOP that we're writing, but in addition to that, there's a whole bunch of Perl scripts on GitHub currently that are being put into a pipeline to automate some of these steps. But yeah, that is just a quick overview of where we are in BGE, at least in the bioscan stream and in some of the workflows that I know. Yeah. I'm happy to take any questions.
That was really good. Yeah, I was going to say there's a variety of where you have your SOPs then and how you store them and questions around versioning of them. But there's a couple. We'll come back to that in the discussion. There's a question from Angela in the chat. I can just read it out. What metadata standard form is used for meta barcoding metagenomics data? What is the source?
Good question. So there aren't really standards. Well, so for barcoding, like single specimen barcoding, there is the barcode of life sort of data standard, which has the fields and the minimum required fields before you can use their database bold. But I'm not aware of anything in the metagenomics or metabarcoding side of things in terms of standards other than the European Nucleotide Archive, like minimum information for a sequence standard. But yeah, that doesn't really capture much of the metadata we do capture.
Angela, did that answer your question?
Yeah, I'm sorry.
Okay.
Yeah, it's like yeah, I would just maybe I could follow up the question, but it's like who does establish those metadata standards? Because I think that's important to know in the future, but perhaps there's another discussion.
We can do that in the discussion. We can, if you
Okay.
pop that into the questions arising from presentation section on the minutes document, we can
Okay,
maybe
fantastic.
revisit it. Great, thanks. There's a couple more questions there, but I think let's put those into the minutes.
Yeah, I can handle
Come back to them. I want to get Caroline talking next.
Cool, thank you.
Thank you, Ben. You all set, Caroline?
I think so. Let's see.
Let's see if you can share. Great. Thanks.
Is that good to you guys? Okay, so my name is Caroline Howard. I'm here just to give you an overview of the SOPs that we use in the Tree of Life core lab team and the ones that are already loaded onto protocols.io. So a very brief background on the Tree of Life. We are a relatively...
Just to stop you, we only see a desktop at the moment.
Ah, okay, hold on. Pause.
Sorry, I was transcribing.
How do I get that? Let me stop sharing.
Maybe you shared the wrong window.
Maybe.
Trick is start the presentation and then do screen sharing.
I think that was what I did, but let me try it a different way. Hold on. Let's do this.
Great. That's it.
Okay. No, go selection. Still
That's
looking good?
working. Yep.
Okay.
Perfect.
Okay, so yes, the Tree of Life is a relatively new program at the Wellcome Sanger Institute in the UK, and we are very heavily involved in lots of projects. They're all listed here, but really the take-home message from this is that we're very invested in generating reference level genome assemblies. So these are beautiful quality genomes, and we make all of these data publicly available as soon as we possibly can during that process. We do that via lots of different projects, and all of these projects cover really the breadth of eukaryotic biodiversity. So really the message here is that we're dealing with lots of different sample types, and my responsibility is to lead the laboratory team. So we need to try and establish some kind of order and some kind of standardization across these thousands of species covering different projects. They all need to come in very different, go through the processes in our laboratory and come out the other end with that same beautiful reference level genome assembly at the end. So the way we do that then is by having these particular protocols that we've developed in the Tree of Life Core Lab. So we've developed this suite of standardized protocols, and these are all available on Protocols.io. Anyone who's interested can scan this QR code and it will take you through to our collection and you'll be able to view all of these. So they are different protocols and they're all here in a modular fashion. So somebody taking a sample from a freezer or from the field and wanting to go all the way through sample preparation, DNA and RNA extraction, fragmentation of DNA, cleanup of the DNA, and then an RNA extraction here, would take one of each of these protocols and put them together as fits the particular sample that you're working on. So we've tried to make this as adaptable as possible so that we can add new protocols all the time. And you'll notice here, particularly we have some that are slightly lighter in color. So these are previous versions. I think this is version 1 and version 2, version 3, and then version 4 in darker color is the one that we're currently using. So that's our method of version control and how we try to give people access to our currently most used, most valuable protocols. And that for us is really the value of Protocols.io, is that it gives us a method by which to share our progress as quickly as we possibly can with anyone who's trying to do this type of work around the world. So for us within the lab, following these standardized protocols means that we're able to progress at pace and with consistency. So even though we're handling lots of different samples that all have different requirements and different needs, we're able to make sure that we apply the same methods and we do improve the standardization and the rigor for each sample as it progresses through our system. And a big part of that, of course, is that we collect the same QC metrics at regular points after each of these protocols and kind of embedded within each of these. So just to take you through some of those protocols, we have a sample prep, triage and dissection. So this is a real decision-making process here when somebody opens a tube in the laboratory and they look at it for the first time and they're trying to decide what to do for the best. And then there's 3 different options of how that sample might be disrupted. You can see there the power masher, Covirus cryoprep, and a bead beater here. We then have a suite of different high molecular weight DNA extraction protocols. And each of these have different pros and cons, and different samples would be used with different SOPs. But one main thing here is that we have both manual and automated versions of most of our protocols. So we have the benefit of a Kingfisher Apex, but then sometimes we also need to make sure that we manually handle samples. Again, there are pros and cons. So we make sure that we have everything available to people. And here is an example of one of our femto traces. That's one of the regular QC metrics that we would capture every time. These are our different fragmentation protocols. We've got the G-tube, which is used for a particular type of library prep, and the megaruptors, which is used for another type of library prep. And then the cleanup technique, which again, we have the manual or the automated procedure. And then the RNA extractions, again, we have a manual or an automated version here. So for any genome that's published, it will be clear exactly which protocol has been used, and people can link back to the exact methodology to understand exactly how we've achieved that reference level genome. So a few examples here. If we were dealing with a mock sample in the lab, for that, the sample would go through this decision-making process. It would be power mashed, go through the automated extraction, and go through these methods here for a PacBio library, and then the automated RNA extraction. However, a grass sample we would deal with slightly differently. So grasses go through the plant bead beating, the automated plant magatract, and then they follow this same process here towards a PacBio library prep and automated RNA. Bivalves, so mollusks can be quite challenging samples to deal with. These tend to be cryo prepped in the lab, automated MagTract, and we would be aiming for standard prep by a hi-fi library here. However, we often find that mollusks can be challenging, so our second backup protocol here would be the mollusk nanobind protocol, which is a slightly different method. Again, for fungi, we look at bead beating and then an automated plant MagTract process. Fungi samples have smaller genomes usually, and they can be quite difficult to fragment, so we would go for a ULI library there. And orchids can be extremely challenging to deal with. So rather than the plant MagTract process, they go through the plant organic extraction, DNA extraction process. So how do we actually use these? Well, we have a few different systems that support us in going through these SOPs and in ensuring that the laboratory work is all carried out to the same standard. So first of all, we have sample selection, which takes place in a system called STS. And before anything is selected, all of the samples must have gone through all of the legal and regulatory compliance, etc. And we have an awful lot of metadata that comes through with every sample. So this is how our STS system looks. This is at the species level. And then once selected a particular species, here we can see all of the tubes that are available to us, and we can make our selection there with all of the relevant metadata. And once we export that sample, it then goes through to benchling. So this is a different view of benchling to one that Ben showed you earlier. This is a particular tissue that has been exported from our STS system into our electronic lab notebook system. And you can see we have a large amount of metadata that comes with that sample. And we're able to put it through sample prep, tissue prep, and the different tasks are all logged here. So these are the different tissue prep methods that we discussed. Each of those are standardized by a particular workflow within the benchling notebooks. And there's different processing depending on the size and the output. And various different factors need to be considered. So again, within benchling, once we've sent those samples into a particular workflow, you can see here that all of these, the disruption methods has been predefined. So that's our scientific manager who selected these particular samples. These are all Lepidoptera. So they will all be power mashed and they will all go through the MagTract extraction process. Similarly, for the DNA and RNA extraction, we have another standardized workflow within benchling, and we can achieve the high throughput by using the Apex. And this is our kind of suite of QC. So within benchling, this is how it looks. These samples have been assigned to Graham. He's gone through the DNA extraction process. Here's the original sample tube here listed. A results PDF from the femto, so that gives us a link through to this trace here. Lots of different metadata are collected. The nanodrop measurements are all in here, qubit measurements. And by having all of these actually worked on at the bench in the lab, there's no transfer required, there's no pen and paper, there's no moving to the office afterwards to transfer your notes. So that's a bit of a whistle-stop tour as to how we are processing things within the Tree of Life. And that's the team who use all of these systems and helped to develop them. So thank you very much.
That was great. Thanks, Caroline. We've got flooding in with questions, I think. So let's take question directed to you. Choose the first one. Okay. To what extent are SOPs based on literature recommendations, and how is that tracked or published themselves as journal articles or similar?
So some of the extraction protocols that we use are, very similar to the manufacturer's recommended guidelines. So if you have a look at our protocols on protocols.io, you'll see that they are linked in the metadata. So the reference link is within the published version then.
So I was going to ask you, so there seem to be a few places where you have, actually, I'm going to stop the recording. Find the button.